Hey guys, okay, so today I'm going to be covering cell types. So we're basically going to be talking about prokaryotes and eukaryotes, okay? So I'm going to go ahead and share my screen with y'all. All right, so I want you guys to go ahead and go to Schoology. You should already be there. So if you go to Unit 3, go to today's folder, January 26th, cell types. This is strange teaching video. That's the video that you're watching right now. So that's me. Okay. So if you go to types of cell information, okay, the first thing that I want you to do today is, okay, I know that there's like 3 videos right here. You only need to watch one of them. This first one, okay? It's the Amoeba sisters talking about prokaryotes and eukaryotes. Okay, the two different types of cells that we have. Prokaryotes are pro for no nucleus. Okay, so basically their DNA is just like free floating, just like a hot mess inside their cell, where eukaryotes are kind of like you and I, okay, where we have membrane bound organelles, we have a nucleus, we have our DNA compacted and nice and like organized really nice. Okay, so it's a 5-minute video, really short. Go ahead and watch this. Okay, and so pause my video, watch the Amoeba sisters, and come back. Okay. Okay, so you've watched the Amoeba sisters. So if you scroll all the way down, again, I'm going to show you how to open up the Note PowerPoint, okay? But because I'm going to basically pull it up on my screen and it's going to be big, you can just pause my video too. So if you wanted to open up the PowerPoint notes, you would click types of cells. It would then download down here to the bottom. I would click it. And there we go. Okay, so life is cellular. You don't have to write that as your title slide. As my title slide, I would just say types of cells. Okay, so next one. Let me get my screen set up. Okay, so the cell theory. So the cell theory has 3 parts. Okay, so instead of these being bullet points, let's go ahead and make them numbers. All right, so the cell theory, first up, all living things are made up of cells. Okay, so Schwann and Sheldon came up with this, and they're the 2 scientists with S names, okay, that we're going to talk about with the cell theory consistently. So Schwann kind of sounds like a swan, right? So that will come back into play. But he said that animals are made up of cells, where Sheldon, the other one that doesn't sound like an animal, came up, said that plants were made up of cells. Okay, so then number 2 for cell theory is that cells are the basic unit of structure and function in living things. So basically what that means is that everything's going to come down to what the heck your cells are doing. And so it's going to come down to what your cellular processes are. These are going to be the basic building blocks of your life. Okay, so then 3 are new cells are produced from existing cells. So Virchow is one of the last scientists that we're going to talk about this unit. And so basically he said that cells are morphing. So this is how we got to see mitosis and meiosis were actually occurring. So we were making new cells from existing cells. Okay, so this is going to, the next couple of slides are basically going to tell you about the scientists that we're learning with the cell theory. Okay, this is going to help you fill out page 3 of your interactive notebook. Okay, so Theodore Schwann, Schwann sounds like swan. Okay, so animal. So his contribution to the cell theory was that animals are made up of cells. So I just want you to write his name and that animals are made up of cells. Okay, so when you got that, next one, Matthias Sheldon. Okay, so Sheldon, you can write his whole name, but I really only care about his last name. Okay, so Sheldon, same thing, contributed to the cell theory. Difference is that he said that Sheldon, plants are made of cells. Schwann, animals are made of cells. Okay, so Matthias Sheldon, plants are made of cells. Okay, then we have Rudolf Virchow. Okay, so he contributed to the cell theory also, and this is where he was saying that cells come from um, already existing cells. Okay, so cells come from cells come from existing cells. Okay, so this is what I want you to write. Rudolf Virchow, his name, and that cells come from existing cells. Okay, so now we're going to talk about prokaryotes and eukaryotes. Okay, so you don't really have to write too much on this slide. The next couple of slides is where you're going to have to do your writing. I just want you to pay attention. Okay, so this is a prokaryotic cell. These two are eukaryotic cells. Okay, so pro means no. Pro sounds like no for no what? No nucleus. So eukaryotes have a nucleus. So see this purple thing? That purple thing's the nucleus. Your DNA is stored really nice and neat and organized inside your nucleus. But prokaryotes, pro for no nucleus, this blue like hot mess that's just kind of like all inside the cell, that's a prokaryotic cell's DNA. So basically, pro for no nucleus means hot mess express for their DNA, where eukaryotes, you are organized, have membrane bound organelles. So your DNA is in a nucleus. Okay, so this the one that my cursor is on right now in the middle is an animal cell, and the one on the right is a plant cell. What's one of the big differences that you can see right now? Okay, you can see the cell wall, okay, in the plant cell. And then in the plant cell, you see this blue like jelly bean looking thing. Okay, we're going to talk about those tomorrow, but this is basically this is the central vacuole, and this is what allows plants to absorb extra water, where animal cells, we don't really absorb extra water. Plants need that. Okay, so going on to the next one. So this slide is basically telling you the differences between a prokaryote and a eukaryote. I would set this up as a T chart in my notes. So I would literally write in my notes, two types of cells equal eukaryote and prokaryote. And then I would do a T chart. So like do a line straight down the middle right here, prokaryotes on the left, eukaryotes on the right, and then list these bullet points. Okay, so prokaryotes are going to be very small, okay, because they don't have a nucleus, they don't have these membrane bound organelles, so they don't need all of this, they don't need the extra size in order to function. Okay, an example of a prokaryotic cell is like bacteria. And then we have eukaryotic cells. So the right side of your T chart is going to be eukaryotes. So eukaryotes are still small. Cells are still itty bitty, okay? Prokaryotes are just smaller than eukaryotes, okay? Eukaryotes, you have a nucleus, okay? You also have membrane bound organelles. So examples of cells like this are going to be plant, animal, fungus, protist, okay? So basically bacteria, prokaryote, eukaryote, plant, animal, fungus, protist, you have a nucleus. Okay, so I know that was a lot. Pause the video if you need a second to get that T chart down. Good news is we're almost done with notes. Okay, so two types of cells. So the going back into it. So this basically your last slide was your T chart where you have prokaryotes and eukaryotes on one side. Now this is going to be like the circle, like your Venn diagram, the similarities in the middle. These are what eukaryotes and prokaryotes both have in common. Okay, they both have DNA. Prokaryotes is just a hot mess and it's free falling in there, where eukaryotes, it's compressed in a nucleus. They both have a cell membrane, okay? They both have cytoplasm. They both have ribosomes, and they both make up living things, okay? I'm not gonna get into the organelles today because we're gonna get into them tomorrow, all right? But tomorrow's video, make sure that you check back because I'm gonna explain all of the organelles really well for you before your test on Thursday, okay? So get this down, pause the video if you need. Okay, so you're done with notes for today. We just have two more slides. Okay, so what type of cell is this? This is a fungal yeast cell. Okay, so first of all, look at my labeled organelles in my picture. It says that I have a nucleus. Okay, so if I have a nucleus, what's going to be stored in there? My DNA. So if I have membrane bound organelles, that I have a nucleus that's storing my DNA, is it prokaryote or eukaryote? It's going to be a eukaryote. All right, last one. It says protist cell, paramecium. So what type of cell is this? Do you see a nucleus? No. Okay, so what type of cell does this have to be? Prokaryote. Okay, so now that was the notes for today. So now go ahead and get out your interactive notebook. Okay, so the first page that we're going to do today is page 3. Okay, so go ahead and get that out. On the paper copy that I passed out to y'all in class, you already have the pictures of the people on there. If you're a virtual student, I need you to literally Google Robert Hooke, take a little picture of him, put it right there. Okay. If you're paper copy, you already have that, so you're good to go. Okay. So I am having you guys fill in the dates for them, but you do not have to memorize the dates. Okay. I just want you to know their inventor's name, specifically, really their last name. That's what you really need to know. And then the description of what they did. Okay. So Robert Hooke, his science was basically founded in 1665. So that's where really he invented the microscope, was around this time period. Okay. So his description, first bullet point is that he built microscopes. Okay. And then he was the first to identify cells. Identify cells. Okay. And he named them. What did he look like? What did he look at under the microscope in order to see that there were smaller holes? He looked at corks. Okay. So that's Robert Hooke. So Robert Hooke built the microscope, first to identify cells using corks. Okay. Antoine van Leeuwenhoek. Okay. Again, butchered his name. Totally fine. You don't have to know how to say it. Just know that Antoine van Leeuwenhoek, or however you want to say it. Okay. He basically was able to view cells at a larger detail, and he was the father of what? He was the father of microbiology. Okay. He basically improved microscopes, which allowed him to view cells in greater detail. Okay. So Matthias Sheldon. Okay. So now we have Sheldon and Schwann, are two S's. Okay. Which one sounds like an animal? Schwann sounds like an animal. So we know he's going to be our, so we can skip down to here. So animals made up of cells. Okay, so we'll go ahead and give his date 1839. Okay, so now we're going to go backtrack to Sheldon. So we know Schwann sounds like swan, so he's animal. So Sheldon has to be the plants. Okay, so he was 1838. So they were like right back to back. Plants made up of cells. Okay, and then last but not least is Virchow. Okay, Virchow was 1855. And what did he say? He was the last part of the cell theory. So he said that all cells come from other cells. Okay, so that's page 3. Okay, that's your cell theory timeline. All right, after you've gotten that done, go to page 9. Okay, so let me insert a text box. Okay, there we go. So now for, we're basically going to be comparing prokaryotic cells and eukaryotic cells. Okay, I'm not going to do all of these with you. You need to give me 3 differences in each one. I'm only going to write 1 in each one. So you need to be listening or going back through your notes. Okay, so prokaryote, pro sounds like no for no what? No nucleus. Okay, where then eukaryotic is what? They have a nucleus, they have membrane bound organelles. Okay, so what So just because a prokaryotic cell doesn't have a nucleus, does that mean that they don't have DNA? No, and they still have DNA, or just remembered it's a hot mess inside the cell. Okay. So there's one characteristic for prokaryotes, there's two for eukaryotes, and then I gave you one similarity for both of them. Okay, so go ahead and fill out the rest of your of page 9. Okay, so what else could you talk about prokaryotic cells? They're smaller than eukaryote. Okay, so prokaryotic are a little smaller, eukaryote are a little larger. So if pro means no for no nucleus, that means that they're also not going to have membrane bound organelles. So that means they're super simple, right? So prokaryotic are simple, eukaryotic are complex. Okay. So what do they both have? We haven't really gotten into it yet, so I'll help you out with this. So they both have cell membranes. Okay, one of them is gonna will sometimes have a cell wall, but both of them will always have cell membranes. Okay, both of them will have cytoplasm. Cytoplasm is the jelly-like fluid that fills the cell. So we'll cover that tomorrow. But there's a couple of things that you could add to your chart. Okay, so then we have one more page to do today. Okay, we're going to label this prokaryotic cell. Okay, so pro sounds like no for no what? No nucleus. Okay, so because it's a prokaryotic cell and it doesn't have a nucleus, what is the DNA? Okay, so DNA is also called a nucleoid. Okay, in prokaryotic cells. I just want you to write DNA. That's fine. So where's my hot mess DNA? It's this big red fuzzy yarn ball that's in the middle. Okay, so changing this to DNA. All right. So prokaryotic cells, in order for them to move, they're going to have these two basically like propellers to help them move. One of them is the flagella. It looks like a big flag. Okay. This is so flagella. Okay, so flagella is like a flag, and it basically acts like a little propeller, just like you would put a boat motor in water, and it would like, and like you just go through the water. Same thing the prokaryotic cell is going to do in the body. Okay, so it's just going to be able to swim using that little propeller. Now, the propeller is used for like super fast movement. Okay, a good example of this is like sperm cells swimming to an egg. They're happening super rapidly, very fast. Right, these little hairy, they kind of look like a beard, scruffy, like mustache. So these little like whiskers, okay, are called pili. And this is for like very slow movement. So like, have you seen that fish like where it like kind of goes really slow with the wave and it's just kind of flat and it just kind of like goes with the motion of the water. So same thing, it's just kind of for like slow movement, and the cell's going to use the pili. Fast movement, the cell's going to use the flagella. Okay. So these little blue dots on the inside, these are ribosomes. Okay. Um, we'll get into the specific function of ribosomes tomorrow. Okay, but both prokaryotic and eukaryotic cells do have them. Okay. The jelly-like fluid that fills the cell is this like blue material that it's probably didn't print out very well on your paper copy, but it's basically going to be all of the air inside the cell. Okay, so cytoplasm. Okay, so on my computer screen, they have color coordinated the different layers of the outer part of the cell, which it's like really nice on color. On your paper copy, not so nice. Okay, so if you have like a color pen or a pencil, I would like y'all to write the name of the vocabulary term up here in the boxes with me, as well as also on your paper, so that way you don't get confused. I did that on my paper copy as well, and it really helped me. Okay, so we're going to start with the outermost layer. Okay, so the tippy top part out here, okay, this is going to be the capsule. Okay, so kind of like the prokaryotic cell looks like a capsule, like a piece of medicine that you would take. All right, so that would be like the outer coating, the protective layer. Okay, then this yellow part that you go that's kind of like the middle part, that's going to be the cell wall. Okay, and then the green part, the innermost part, is the plasma membrane. Plasma membrane, which is also the cell membrane. Okay, so the only one that we haven't answered yet is this one, and this is a plasmid. Okay, a plasmid, we'll cover that tomorrow too, but I really want you to know all of the other ones first. Okay, we're not going to test you on plasmid. We're really going to test you on you knowing if a prokaryote has a nucleus or not, what a flagella, pili, plasma membrane, and cytoplasm is. Okay, so that is it for today for notes and notebook pages. So if you have any other questions, please shoot me an email or message me on Schoology. I miss you guys so much, and I hope you guys are having a great day. Bye, guys.